All right. So we now turn our attention to the synthesis and the metabolism of cholesterol as well as steroid. Now, cholesterol perhaps is one of the most defamed organic molecules or biologically important molecules. Our our perception of cholesterol is that of of a culprit, right? In many instances. But cholesterol is actually an important component of cell membranes, as you've already learned about in the past meetings. The negative perception of cholesterol is primarily attributable to the correlation of cholesterol with incidence of cardiovascular diseases. That is definitely a serious concern. But we have to realize that cholesterol is an important component of living systems. Their role in maintaining the structural rigidity and integrity of the cell membrane of mammalian systems is just one of the many important functions that they perform. Another important function, for example, would be the fact that cholesterol serves as a precursor of bile acids and bile salts, and that important molecules like vitamin D, for example, are in one way or another derived from cholesterol. Okay? Alright, so in this discussion, we're going to focus on cholesterol and steroid metabolism. Now, I've already discussed in one of the lecture videos that I shared with you the anabolism of lipids or the biosynthesis of fatty acids. So I'm not going to touch on that anymore. What I'm going to do is I'm going to focus primarily on cholesterol, its biosynthesis, as well as the fates of cholesterol, which will naturally entail discussion of lipoproteins. Okay? Alright. So as I've mentioned, cholesterol is doubtless the most publicized lipid, right? Notorious because of the strong correlation between high levels of cholesterol in the blood in the incidence of human cardiovascular diseases. Less well advertised, as I mentioned earlier, is cholesterol's crucial role as a component of cellular membranes and as precursor of steroid hormones and bile acids. I've already mentioned this earlier as well. So, cholesterol is an essential molecule in many animals, including us, humans, but it is not required in the mammalian diet. Why? Because all cells can synthesize cholesterol from simple precursors. Okay, so the structure of this 27 carbon compound suggests a very complex and complicated biosynthetic pathway. Right? Look at the structure of cholesterol here, and you're going to notice that it has a complicated structure. It has one, two, three, four rings, three six-membered rings, one five-membered rings. There's an aliphatic chain connected to ring D, and there's a hydroxyl group found in ring A. Now, despite its complexity and the anticipated complicated biosynthetic pathway that leads to the synthesis, to the biosynthesis of cholesterol. Remarkably, cholesterol is actually derived from acetate, right? All of the carbon atoms that you see here are derived from acetate, okay? All of its carbon atoms are provided by a single precursor, acetate. Okay, now the acetate, of course, that cholesterol or that serves as precursor for cholesterol biosynthesis is acetyl-CoA, not acetate itself, but acetyl-CoA and related molecules. Now, the isoprene here, structure of the isoprene is shown here, which in systematic organic IUPAC nomenclature is 2-methyl-1,3-butadiene. All right, 2-methyl-1,3-butadiene or isoprene are essential intermediates in the pathway from acetate to cholesterol. Isoprene are also serve as precursors to many other natural lipids. And the mechanisms, by which isoprene units are polymerized are similar in all of these pathways. Okay, meaning synthesis of cholesterol and other natural lipids derived from isoprenes have or share similar steps or similar pathways. All right. Okay. Now, as I mentioned earlier, cholesterol is actually synthesized from acetyl-CoA. All right. Okay. So cholesterol is made from acetyl-CoA in 4 stages, and this is like a deja vu of sorts, because we've already encountered the case of synthesis of fatty acids from acetyl-CoA, right? So this time around, we're encountering the use of acetyl-CoA as precursor for a different lipid, which is cholesterol, right? So cholesterol, like long-chain fatty acids, is made from acetyl-CoA. Now, the synthesis takes place in 4 stages. The first is a condensation of 3 acetate units to form a 6-carbon intermediate called mevalonate. All right, so here it is. All right, I hope that you can see my cursor here. So acetate, as you can see here, 3 molecules of acetate are condensed to form the molecule mevalonate. That's the first stage. The second stage entails the conversion of mevalonate to activated isoprene units. So this is an example of an activated isoprene unit. So you have here a pyrophosphate that is linked to the isoprene unit. Okay. Now, the third step involves the polymerization of 6 5-carbon isoprene units to form the 30-carbon linear squalene molecule. Now, 6 times 5, that's 30, right? So that's why the linear squalene or the squalene molecule is 30 carbon atoms, because it comes from the polymerization of 6 5-carbon isoprene units. So this is squalene, so from activated isoprenes to squalene. Then the fourth step is the cyclization of squalene, here, the cyclization of squalene to form the 4 rings of the steroid nucleus, with further series of changes like oxidation, migration of methyl groups to produce cholesterol. So the 4th stage in the biosynthesis of cholesterol, again, is the transformation of squalene to cholesterol, or the cyclization of squalene to cholesterol. Okay, now let us examine these 4 stages in detail. First stage in cholesterol biosynthesis leads to the intermediate mevalonate. All right, so as you can see in this case, 2 molecules of acetyl-CoA react with each other by the reaction being catalyzed with the enzyme thiolase to produce acetoacetyl-CoA. Now, this is an example of a Claisen condensation. Do you remember back in organic chemistry, you learned about the reaction between 2 esters under basic conditions to produce beta-ketoester? Now, the biological, the biological version or the biological counterpart in such organic reaction in the lab occurs in this case, not involving esters, but thioesters. Okay, so 2 molecules of acetyl-CoA or 2 thioester molecules react in a Claisen condensation to produce beta, remember this is alpha, beta keto thioester. This is acetoacetyl-CoA. Now, the enzyme hydroxymethylglutaryl CoA synthase catalyzes the reaction between another molecule of acetyl-CoA and acetoacetyl-CoA. What happens in this case is actually, if you're going to notice that this is not a Claisen condensation, this is actually an aldol addition reaction, in which case this methyl group, all right, has been deprotonated and acts as an aldol. It's a nucleophile that attacks this carbon atom of the carbonyl group to, in the process, produce beta hydroxy beta methyl gluteryl CoA as intermediate. Of course, I want you to notice that the coenzyme A at this carbon atom, this coenzyme A, has been hydrolyzed. That's why you end up releasing another molecule of coenzyme A. All right, so this is acetyl CoA, 3 molecules of acetyl CoA essentially condensing to form beta hydroxy beta methyl gluteryl CoA. Alright, so what enzyme catalyzes that? HMG CoA synthase. But is that the end of the stage 1 of the biosynthesis of cholesterol? No. Beta hydroxy methyl gluteryl CoA will need to be transformed into mevalonate. And how does that happen? Well, the enzyme hydroxymethyl gluteryl CoA reductase comes into the picture, and as you're going to notice, the enzyme utilizes 2 molecules of nicotinamide adenine dinucleotide phosphate, NADPH. All right. Okay, so what do you think happened in that case? What happened is that this coenzyme A has actually been removed or displaced, and this group is actually reduced. Right. The C double bond, I'm sorry, I'm going to go back to that. So the C double bond O here has been converted into CH2OH, right? And the coenzyme A has been released. That's why you end up with mevalonate, right? Now this third reaction is the committed and the rate limiting step in the process, or in stage 1 of cholesterol synthesis. Actually, it's the rate limiting step in the biosynthesis of cholesterol. That's why many drugs that lower the amount of cholesterol target HMG CoA reductase as the target enzyme, right? Because that's the committed step, the rate determining or the rate limiting step in the synthesis of cholesterol. Okay, now so the HMG CoA reductase, an integral membrane protein in the smooth endoplasmic reticulum, is the major point of regulation on the pathway to cholesterol. This is a recurring theme in metabolism. The slowest step in the mechanism, the rate limiting step, the committed step, is the point of regulation or the point of control for the pathway. Okay, all right, so now we have mevalonate. What is the second stage of the synthesis of the biosynthesis of cholesterol? The conversion of mevalonate to 2 activated isoprenes. Okay, so I want you to look at this structure. So this is mevalonate. What happens here is that mevalonate reacts with a molecule of adenosine triphosphate to, in the process, produce 5-phosphomevalonate, right? 5-phosphomevalonate. Now, what do you notice? So the reaction between mevalonate and ATP causes the phosphorylation of this hydroxyl group, right? The process to produce 5-phosphomevalonate. Now, the enzyme that catalyzes this reaction is mevalonate 5-phosphotransferase. Now, this requires 1 molecule of ATP. Now, the second reaction is further phosphorylation of the 5-phosphomevalonate to, in the process, produce 5-pyrophosphomevalonate, right? Now, this is catalyzed by the phosphomevalonate kinase. Phosphomevalonate kinase, right? Okay, now the third step requires another molecule of ATP. Now, I want you to notice that the group that is phosphorylated is actually this hydroxyl group, the remaining hydroxyl group. So you end up producing 3-phospho-5-pyrophosphomevalonate. Okay, now this is catalyzed by pyrophosphomevalonate decarboxylase. Now, I want you to look at the reaction. Now, notice that there is a bracket here. Now, the bracket indicates that that substance is like an intermediate that is formed at the active site, but is not actually the final product of the reaction. What's going to happen is that this carbon or carboxylate group will leave the molecule in the form of carbon dioxide. So the pair of electrons will delocalize to this carbon, which will further delocalize to this carbon atom, causing the breakage of the carbon-oxygen bond of the phosphate group. In the process, releasing carbon dioxide and inorganic phosphate. So you end up forming delta-free isopentenyl pyrophosphate, this structure. Now, delta-free isopentenyl pyrophosphate is actually then or afterwards isomerized to dimethyl allyl pyrophosphate. Right? See here, dimethyl allyl pyrophosphate. Okay. Now, these are the two activated isoprenes that we're talking about. So here, we have seen the transformation of methylamine into two activated isoprene units. Now, they're activated because you have here a very powerful or a very good ligand, right? Pyrophosphate, right? Now, the third stage in the synthesis of cholesterol is the condensation of 6 activated isoprene units to form squalene. So you have here dimethyl allyl pyrophosphate and delta-3 isopentenyl pyrophosphate. I'm just going to refer to it from now on as isopentenyl pyrophosphate. Now these two molecules react to form geranyl pyrophosphate. So I want you to notice that isopentenyl pyrophosphate and dimethyl allyl pyrophosphate undergo a head-to-tail condensation. Now, as I mentioned here, the head is the end to which the pyrophosphate is linked. So this is the head, that is the tail, that is the head, and this is the tail. So head to tail rather, head to tail condensation. If you're going to notice, this part, okay, this one, can I write here? I'm not sure if I can, maybe I can underline, strike through, draw. Okay, maybe I can draw. So here, this part, it's too dark. Okay. Okay, so it was too. Is there a way for me to control? It's too. Okay, so what I'm trying to say is that this part, that part, actually came from this part, right? Of dimethyl allyl pyrophosphate. While this part it's actually this, the isopentenyl pyrophosphate. So it's a reaction. Essentially what happened here is that a carbocation is produced when the pyrophosphate departs the molecule, and that carbocation acts as an electrophile that attacks this double bond, in the process generating a carbocation a positive charge here, which is neutralized by losing a proton or a hydrogen atom from this carbon atom. That's why the double bond is formed here. Okay, I hope that that is clear. So this is clearly an extension of what you've learned in organic chemistry. Okay, so in the process you end up synthesizing geranyl pyrophosphate. Now, geranyl pyrophosphate then undergoes another head to before anything else. This is now 10 carbon atoms, right? 10 carbon atoms long. Okay, so 5 plus 5, 10. Alright, so the next step is when geranyl pyrophosphate undergoes another head-to-tail condensation with isopentenyl pyrophosphate, yielding a 15 carbon intermediate called farnesyl pyrophosphate. So again, isopentenyl pyrophosphate. So what happens? Now this part of geranyl pyrophosphate is actually this. That one. Okay. Now that is going to become a carbocation which is going to attack the isopentenyl pyrophosphate, so that in the end, again, as before, a positive charge will develop here, which will be neutralized by loss of a proton from this carbon atom. So this part, okay, this part actually came from isopentenyl pyrophosphate. Okay, I hope that is clear. All right, so we now have 15 carbon atom intermediate called farnesyl pyrophosphate. Now, the next step, remember, the final step and stage, the final product, I mean, not the final, but the end product in stage 3 is squalene, right? So what happens here is that two molecules of farnesyl pyrophosphate join head to head with the elimination of both pyrophosphate groups. So as you're going to notice, this is, I'm sorry, that is one farnesyl. That's one farnesyl, and the other one is again another farnesyl. All right, they were linked to each other this time around, head to head. Okay, now this happens in an enzyme, and you will know in an enzyme active site, and you will notice that it's actually a reductive reaction because it entails the consumption of nicotinamide adenine dinucleotide phosphate. Okay, all right. So now that we have the squalene, we're ready for stage 4. All right. Stage 4 of the process is the conversion of squalene to the 4-ring steroid nucleus. Now, here's the steroid nucleus that I'm talking about. You will notice that you have there 4 rings. Let me change this into, I just want the point. So point, select. Okay, so this one. So, A, B, C, and D rings. The A, B, and C and D rings are all fused together. A, B, C rings are cyclohexanes, while the D ring is a cyclopentane. Okay, so you will notice here that we have written or we have drawn the structure of squalene in this particular manner. All right. Now, why did we draw it in that way? In order to make the results of the cyclization step or cyclization steps more apparent to you. So for example, what happens first is that squalene monooxygenase epoxidizes carbon 2 and 3. So you end up here with an epoxide. Now this is going to be opened up, generating a positive charge on carbon number 2. So this carbon atom will then attack carbon number 2, generating a positive charge here, which will again be attacked by this carbon atom. So end up forming 2 rings. A positive charge will be formed here, which will be attacked by this carbon atom, forming the 3rd ring. Now, a positive charge develops here, which will be attacked by this carbon atom, forming the 4th ring. Right? So in the end, what you get is the this intermediate. Now, this intermediate undergoes further reaction in order to produce lanosterol. Right? This is what we have in mammalian, in animals, essentially. We have lanosterol. Okay? Now, it is important to highlight that the double bonds of the product squalene 2,3-epoxide are positioned so that a remarkable concerted reaction can occur and can convert the linear sequence of the linear squalene epoxide to a cyclic structure. All of these ring closing reactions happen at the same time. All right, so you can imagine the enzyme contorting or twisting the molecules in twisting the molecule of squalene epoxide in this particular arrangement so as to encourage the reaction, so as to guide the carbon atoms into reacting in this particular manner in order to produce the intermediate for lanosterol. Okay, now stigmasterol, it's produced in plants. Ergosterol, it is produced in fungi. Okay, in mammals, what we have is lanosterol. Okay, now Lanosterol in um, mammals, as I've mentioned, is finally converted to cholesterol in a series of about 20 more reactions. 20 more reactions would include the migration of some methyl groups and the removal of the others. All right, so here, after multiple steps, 20 steps, lanosterol is converted into cholesterol. All right, lanosterol is converted into cholesterol. So here are the individuals who largely contributed to the elucidation of this remarkable and amazing pathway, cholesterol biosynthesis. You have Conrad, Bloch, Theodore Leinen, John Cornforth, and George Popchak. Okay. So cholesterol is the sterol characteristic of animal cells, as I've already mentioned. So plants, fungi, and protists make other closely related sterols instead. Okay. Now they use the same synthetic pathway as far as choline 2,3 epoxide, at which point the pathways diverge slightly, yielding other sterols, in this case to produce stigmasterol, and in the case of fungi, you end up with ergosterol. Okay, all right, okay. So, as I've mentioned, cholesterol is an important biological molecule. So, what are the metabolic fates of cholesterol? So, again, I want you to pay attention to the structure of cholesterol. Okay, much of the cholesterol synthesis in vertebrates actually takes place in the liver. I'm not sure if I've mentioned it already, but you can liken the liver to the brain. Like brain is to the nervous system, is liver is to the um, metabolic system, all right, or the metabolic landscape, because it's the liver that actually takes care of for example, maintaining blood glucose levels, ensuring that peripheral tissues, muscles, heart, kidney have their sufficient share of fuel, etc. Okay, so it's the liver that does the heavy lifting in ensuring that all of the tissues are provided with their energy source. Okay, so let's go back to cholesterol. Much of the cholesterol synthesis in vertebrates takes place in the liver, right? A small fraction of the cholesterol made there is incorporated into the membranes of hepatocytes. Remember, hepatocytes are liver cells. So as we've already mentioned earlier that um, cholesterol is an important component of the cell membrane. It's important for maintaining the rigidity of the cell membrane. So a small fraction of the cholesterol synthesized in the liver is actually incorporated into the liver cell membrane. Okay, but most of the cholesterol is exported in one of three forms. End up with biliary cholesterol, bile acids, or cholesterol esters. I'm sure you're familiar with bile salts, all right? Bile acids. These are, I asked one of my students before because I forgot the organ that actually releases this. It's gallbladder, right? So it releases bile acids and bile salts. These are like surfactants. These are cholesterol derivatives that have negative charges, for example, that aid in the emulsification of fats and are therefore necessary for digestion, right? Okay. These are, as I've mentioned, bile acids and their salts are relatively hydrophilic cholesterol derivatives that are synthesized in the liver and aid in lipid digestion. Okay. Now, so that's one possible fate of the cholesterol that is synthesized in the liver. Okay, and remember, much of the synthesis of cholesterol occurs in the liver. Now, I've mentioned here another fate, cholesterol esters, right? So the cholesterol that is synthesized in the liver can actually be transformed into an ester. Cholesterol, for example, can react with fatty acyl-CoA, right? In the process, producing a cholesterol ester. Okay? All right, so the cholesterol ester that is produced from fatty acyl-CoA and cholesterol is relatively hydrophobic, right? As you can see here, there's no more any hydroxyl group. The hydroxyl group is the only polar group in cholesterol, but this time, after esterification, it's already been masked into an ester, making the molecule predominantly nonpolar. Couple that to the fact that fatty acyl group has a relatively long alkyl chain, right? So this molecule, cholesterol ester, is quite nonpolar. So the enzyme that catalyzes the formation of cholesterol ester is the enzyme called acyl-CoA cholesterol acyltransferase, or ACAT. All right. So this enzyme, as I've already mentioned, catalyzes the transfer of a fatty acid from coenzyme A to the hydroxyl group of cholesterol. Okay. So this cholesterol esters are very important because this are predominantly the form by which cholesterol are transported to different tissues via the lipoproteins. Okay, now ask yourselves the question, what are lipoproteins? Okay, so cholesterol and cholesterol esters, like triacylglycerols and phospholipids, are essentially insoluble in water. Yet, as we already know, they must be moved from the tissue of origin, liver or intestine, to the tissues in which they will be stored, like adipose tissues, or consumed, like muscles. So, how do we accomplish that? They are carried in the blood plasma, right? In the blood plasma as plasma lipoproteins. Now, the plasma lipoproteins, these are, can think of them as they're not vesicles. They are, I'm sure you've heard me mention before micelles. These are micellar assemblies, right? In which the hydrophobic tails, of the phospholipids are projected inward, while the hydrophilic heads are projected outward, interacting with the molecules of water. Okay? Okay. So the lipoproteins, micellar assemblies, you can think of them as um, transports, right? They are the means by which cholesterol and cholesterol esters are transported to different tissues. As I mentioned here, they are carried. As we mentioned here, they are carried in the blood plasma as plasma lipoproteins, which are macromolecular complexes of specific carrier proteins, apolipoproteins. These are carrier protein, apolipoproteins with various combinations of phospholipids, cholesterol, cholesterol esters, and of course, triacylglycerol. Okay, so as you can see in this diagram, we have here the phospholipid monolayer, which the polar heads are exposed to water while the nonpolar tails are projected inwards. All right. These nonpolar tails are the ones that interact with the nonpolar triacylglycerol, cholesterol, and cholesterol esters. And we have here an apolipoprotein. Apolipoprotein, it's a carrier protein whose importance in this role will become more apparent later. Okay, so that's why we call them lipoproteins, because they are assemblies of lipids, triacylglycerols, cholesterol, cholesterol esters, and phospholipids, and of course, proteins, the apolipoproteins. So call them lipoproteins. All right. So, the lipoprotein particles are classified according to their sizes and to their densities. As you can see here, they are roughly spherical complexes with hydrophobic lipids in the core and the hydrophilic amino acids, as well as hydrophilic heads of phospholipids at the surface. You're most familiar, I believe, with the classification wherein they are called bad and good cholesterol. So chylomicrons, VLDL, IDL, LDL, these are the bad or the non-HDL cholesterol, while the good cholesterol is actually the high-density lipoprotein. The high-density lipoprotein is the densest, it is the smallest, right? While the um, lowest density would be the chylomicrons, it's the biggest, followed by VLDL. And these are the bad cholesterols, a bad cholesterol. VLDL, IDL, LDL. Okay, so different combinations of lipids and proteins produce particles of different densities ranging from chylomicrons to high-density lipoproteins. Now, these particles are actually separable by ultracentrifugation and can be seen or visualized by electron microscopy, as you can see in this case. Chylomicrons here are huge, right? VLDL followed by LDL and then of course the HDL. Now each class of lipoprotein has a specific function which is determined by its point of synthesis, where it's been synthesized, lipid composition, how much acylglycerol, how much cholesterol ester, and of course apolipoprotein content. So here's a table showing us the major classes of human lipoproteins and some of their properties, like the composition in weight percent, the density, and the amount of protein, phospholipids, free cholesterol, cholesterol esters, and triacylglycerol. So chylomicrons are the least dense, HDL are the densest, and you will notice that the one which has the highest percentage of proteins are the HDL, all right, 55% in weight percentage, while the chylomicrons have the highest percentage of triacylglycerol. So the proteins are the dense part of the lipoproteins. See here are just another way of visualizing the lipoproteins. Okay, so at least 9 different apolipoproteins are found in the lipoproteins of human plasma. They are distinguishable by their size, their reactions with specific antibodies, and of course their characteristic distribution in the lipoprotein classes. As you can see here, tabulated in this table, the different apolipoproteins, their molecular weights, and their lipoprotein association, are they found in HDL, chylomicrons, and of course their function, if it is known. For example, in ApoA1, it activates LCAT, interacts with ABC transporter. We're going to learn exactly what LCAT is in a moment. Now, these protein components, the apolipoprotein, acts as signals targeting lipoproteins to specific tissues or activating enzymes that act on the lipoproteins. It's important to take note of that because remember, lipoproteins serve as vehicles. They deliver the cholesterol and cholesterol esters to the target tissues. So lipoprotein and the apolipoproteins serve as the the molecule that allows it to recognize the tissue for which it is intended to go, okay, or to which it is intended to go. Okay, so let's talk about the chylomicrons. The chylomicrons are synthesized in the endoplasmic reticulum of epithelial cells that line the small intestine. So the origin of the chylomicrons then is intestinal mucosa, right? I mean intestine. So then move through the lymphatic system and enter the bloodstream. The apolipoproteins of chylomicrons include ApoB48, ApoE, and ApoC2. Now, ApoC2 activates lipoprotein lipase in the capillaries of adipose, heart, skeletal, and lactating mammary tissues, allowing the release of free fatty acids to this tissue. So what is important to take note is that ApoC2 activates a lipase, right? And lipases, if you will remember in our earlier discussion, are enzymes that cleave triacylglycerols or triglycerides into fatty acids and glycerols. So, for example, ApoC2 here, once it activates the lipase in adipose, heart, skeletal muscle, and lactating mammary tissue, so that fatty acids will be released and made available to those particular tissues. Okay, as a source of energy, perhaps, or in the case of adipose, for storage purposes. Now, chylomicrons thus carry dietary fatty acids. It is important to take note of that. Dietary fatty acids to tissues where they will be consumed or stored as fuel. Right? This is just a cartoon representation of what chylomicron look like. It's one huge, giant globule. Okay. Now, the remnants of the chylomicrons, which have been depleted most of their triacyl glycerols but still contain cholesterol, ApoE and ApoB48, move through the bloodstream to the liver. So ultimately, its final destination is the liver. Of course, after it has delivered its triacylglycerol cargo from the intestine to the target tissue, like adipose, heart, lactating mammary tissues, etc. All right, so the receptors in the liver bind to the ApoE in the chylomicron remnants and mediate their uptake by endocytosis. All right, in the liver, the remnants release their cholesterol and are degraded in the lysosomes. So the liver, in this case, essentially collects the excess or the cholesterol that has been that has been carried by the chylomicrons. Okay, so those are the chylomicrons. Let's now turn our attention to the VLDL, or the very low density lipoprotein. Okay, cartoon representation is shown here. So when the diet contains more fatty acids than are needed immediately as fuel, in other words, say for example, when you eat lechon or you eat barbecue and you consume the fat part, the fatty part, they're converted to triacylglycerols in the liver and packaged with specific apolipoproteins into very low density lipoproteins. Again, when you eat a lot of fatty acids, they are converted to triacylglycerols in the liver. But these triacylglycerols have to be delivered to adipose tissues, right? Okay, so it is important to take note of that excess carbohydrates, can be transformed into fatty acids, right? Because when you eat a lot of carbohydrates, like a lot of cakes, a lot of rice, you end up with a lot of acetyl-CoA. And acetyl-CoA, in our previous discussion, is the precursor for the synthesis of fatty acids. So essentially, excess carbohydrates are transformed into fats, right? Okay, so excess carbohydrate in the diet can also be converted to triacylglycerols in the liver and exported as VLDLs, all right? So in addition to triacylglycerols, VLDLs contain cholesterol and cholesterol esters, as well as ApoB100, ApoC1, ApoC2, ApoC3, and ApoE, okay, apolipoproteins. Now, these lipoproteins are transported in the blood from the liver, remember, from the liver to muscle and adipose tissue. To muscle for consumption and adipose tissue for storage, where activation of the lipoprotein lipase by ApoC2 causes the release again of free fatty acids from VLDL to the release of fatty acids from VLDL triglycerides or triacylglycerols. So the adipose sites take up the fatty acids, reconvert them to triacylglycerols, as I mentioned earlier, for storage, and store the products in intracellular lipid droplets. Most VLDL remnants, in other words, those which have lost their triacylglycerol, are removed from the circulation by hepatocytes, again by the liver, right? Now, the uptake, like that for chylomicrons, is receptor mediated and depends on the presence of ApoE in the VLDL remnants. Okay, so again, the ApoE is important for being recognized by the targeted tissues, or the target tissues, or the tissues of ultimate destination. Okay, Now, the high density lipoproteins, these are the good cholesterol, right? I think skipped. So, low density lipoproteins first. The loss of triacyl glycerol converts VLDL to VLDL remnants. We also call them intermediate density lipoproteins, all right? So you see here another cartoon representation of a low density lipoprotein this time around. So further removal of triacylglycerol from VLDL produces low density lipoprotein. All right, these are very rich in cholesterol and cholesterol esters, right? Because typically what is unloaded to the muscles and to the tissues are just the triacylglycerols, right? Of course, some cholesterols and cholesterol esters are lost, but it's not as much as the amount of triacylglycerols or triglycerides that have been unloaded to adipose tissues and other peripheral tissues. So in the end, the lipoprotein becomes enriched in cholesterol and cholesterol esters, right? So very rich in cholesterol and cholesterol esters and containing ApoB100 as their major apolipoprotein. LDLs essentially carry cholesterol to extrahepatic tissues that have specific plasma membrane receptors that recognize ApoB100. So the low-density lipoproteins can be thought of as trucks that deliver cholesterol and cholesterol esters to peripheral tissues, okay? To muscles and to other tissues, for example, that require cholesterol, okay? Now, I mentioned ApoB100. These receptors mediate the uptake of cholesterol and cholesterol esters into the cells, okay? So again, you can imagine the LDLs as the delivery trucks that transport cholesterol and cholesterol esters to peripheral tissues. Now, the high-density lipoproteins, this originate in the liver and small intestine as small protein-rich particles that contain relatively little cholesterol and no cholesterol esters, okay? So from the liver, from the small intestine, no cholesterol esters, okay? Now, HDLs contain ApoA1, ApoC1, ApoC2, and other apolipoproteins, as well as the enzyme lecithin cholesterol acyl transferase, LCAT. I mentioned earlier that we're going to encounter this. So, what is LCAT for? It catalyzes the formation of cholesterol esters from lecithin, which is phosphatidylcholine and cholesterol. Okay, so you see here, phosphatidylcholine and cholesterol, all right, are used as the reactants for the synthesis of cholesterol ester. Lecithin cholesterol acyl transferase catalyzes the formation of cholesterol esters from lecithin and cholesterol. Okay, all right. Now, the LCAT on the surface of nascent, newly forming, when you say nascent, it's newly forming HDL particles, convert the cholesterol and phosphatidylcholine of the chylomicron and the VLDL remnants. Remember, the VLDL remnants, these are the VLDL that are now devoid or at least significantly that lost significant amounts of triglycerides. So the LCAT on the surface of nascent HDL particles convert the cholesterol and phosphatidylcholine of chylomicrons and VLDL remnants to cholesterol esters, which begin to form a core in the interior of the HDL, transforming the disc-shaped nascent HDL to a mature spherical HDL. All right. So this time around, the cholesterol-rich lipoprotein then returns to the liver, where the cholesterol is unloaded. Some of this cholesterol is converted to bile salts. We've already encountered bile salts earlier. So do you now understand, while HDL is called the good cholesterol, It's called a good cholesterol because essentially it brings cholesterol to the liver. Remember, LDL, VLDL, IDL, these are the bad cholesterol because in the process of carrying cholesterol from the liver or from the intestine to the peripheral tissues, some cholesterol or cholesterol esters um, may fall along the wayside, meaning it may what do you call that? It may by accident be unloaded to the blood vessels, all right, which lead to the formation of atherosclerotic plaques, which is of course associated with cardiovascular disease. Now, that's why they're the bad cholesterol. The good cholesterol is the HDL because it collects cholesterol from the peripheral tissue environment, specifically from the chylomicron and the VLDL remnants, back to the liver. So earlier I mentioned that LDL, HDL, chylomicrons, you can think of them as trucks, right? That deliver cholesterol and triacylglycerol to different tissues. Cholesterol falls by the wayside, but you can think of HDL as street sweepers that clean the road, the blood vessels, for example, and collect the cholesterol, right? And then returns them to the liver. That's why they are the good cholesterol, okay? They collect the cholesterol and make sure that the probability of cholesterol depositing in the blood vessels is minimized. Okay. Now, as I mentioned earlier, the chylomicrons and the VLDL originate from the intestine and the liver respectively. They go through the blood vessels, the capillary, for example. The chylomicron remnants, the VDL remnants, and the LDL remnants go back to the liver. Remember, the HDL here essentially collects the cholesterol from the remnants of the VLDL and the chylomicron and brings it back to the liver, the cholesterol and the cholesterol esters. Now here's a very interesting figure where you can see the blood plasma samples that are collected after a fast and after a high-fat meal. You can see it's cloudy. The blood plasma is cloudy after a high-fat meal because of the large amount of lipoproteins, the triacylglycerols that are carried by these lipoproteins. Remember, lipoproteins are colloidal particles which make the suspension cloudy. Okay, now I did not mention it earlier, but the HDL is associated with reverse cholesterol transport because it transports cholesterol from extrahepatic tissues to the liver. Remember, HDL originates from the liver and the intestine and collects cholesterol from extrahepatic tissues and deliver it back to the liver. Okay? All right. Now, this is essentially endocytosis, as I mentioned earlier, like the chylomicron remnants. Ultimately, right, it's internalized by the liver. So here, an LDL particle is going to be recognized by the cell, for example, that it targets, for example, the liver. All right, the cholesterol ester will be internalized via receptor-mediated endocytosis. So LDL, ApoB100, which is recognized by the liver cell, for example, or the hepatocyte, and you end up forming an endosome. Okay, it's like the lipoprotein has been engulfed, right? So the endosome is formed, and then it's essentially digested, the cholesterol ester is released, and the other contents, of course, of the LDL particle. Okay, now the cholesterol biosynthesis is regulated at several levels, right? So you have here the cytosol, right? Cytosol. Cholesterol biosynthesis occurs, remember, in the cytosol, right? So you have here the endoplasmic reticulum, okay? And you have here the SREBP. It's an association, an SREBP protein and an SCAP protein, right? These are the proteins that are associated with each other. Now, sterol, it binds to the SCAP and it prevents the release of SREBP, right? So essentially, this assembly has to migrate to the Golgi complex. But in the presence of the sterol, it does not do so. Okay, it binds to SCAP and it prevents the release of SREBP. So let's assume that there's no such sterol. The SREBP SCAP assembly is able to migrate from the endoplasmic reticulum in the cytosol to the Golgi complex. The SCAP SREBP assembly is cleaved by a protease, so you release the SREBP and you end up here with you release the SREBP, yes, because it's cleaved here, right? Another protease cleaves this portion of the SREBP, okay? And what happens is that this domain of SREBP then migrates to the nucleus, which then targets the genes for cholesterol biosynthesis, for example, okay? Now, in the case of or when a sterol is present, it binds to the SCAP, right? Binds to the SCAP and it prevents the release of SREBP. In other words, this SREBP domain is not released, and as a result, the transcription of genes associated with the synthesis of cholesterol is, of course, not activated, right? So this just tells us that the synthesis of cholesterol is regulated at the level of transcription, okay? I think I mentioned it before that the cell or the living system essentially prepares itself for a metabolic pathway by synthesizing enzymes necessary for it. For example, the moment you eat lunch, the genes coding for the enzymes of glycolysis and the citric acid cycle will be activated so that its gene products, the enzymes for glycolysis and citric acid cycle, will be formed or produced and will be used for the synthesis, or rather for the oxidation of glucose. So the same is true in this case. And what happens in this case is that the enzymes necessary for the transcription of target genes, I'm sorry, the enzymes that are coded for by the genes for cholesterol biosynthesis. Remember the different enzymes that we talked about earlier? There are many, right? In the different stages of cholesterol biosynthesis. These are activated by the migration of the SREBP domain to the nucleus. Again, so SREBP SCAP will migrate from the endoplasmic reticulum in the cytosol to the Golgi complex. It's going to be cleaved by a protease, another cleavage by a different protease, will lead to the release of this SREBP domain, which leads to the activation of target genes, the genes that are responsible for the synthesis of cholesterol. The presence of sterol, such migration is essentially what happens is the migration is inhibited, and that the binding of the sterol prevents the release of SREBP. Okay. Okay. Now, cholesterol biosynthesis, as I've mentioned, is regulated at several levels. So, the regulation of cholesterol formation balances synthesis with the dietary uptake. Okay, so glucagon, for example, promotes phosphorylation or the inactivation of HMG-CoA reductase. Remember, that's the rate-limiting step, the committed step in biosynthesis of cholesterol. So glucagon promotes its phosphorylation and its inactivation. In other words, when glucagon levels are high, the cholesterol biosynthesis is inhibited, while insulin promotes the dephosphorylation of HMG-CoA reductase and it activates it. Okay, now see here, acetyl-CoA to beta-hydroxy beta-methylbutyryl-CoA to mevalonate. Glucagon inhibits that step, insulin activates it. What else? The interesting thing to note is that there is an unidentified metabolite or metabolites of cholesterol that, as mentioned here, stimulate the proteolysis of HMG-CoA reductase. So this intermediate is, well, I'm not sure if they were already able to identify it, but this is an unidentified metabolite, right, that essentially inhibits cholesterol biosynthesis, right? Okay, it hydrolyzes HMG-CoA reductase, which leads to the lowering in the rate of cholesterol biosynthesis. It also inhibits the receptor-mediated endocytosis of cholesterol from LDL cholesterol. Okay. Now, I want you to notice that cholesterol here actually encourages or enhances the activity of ACAT. All right, it's the enzyme that is responsible for the synthesis of cholesterol esters, right? Okay. All right. Now, as I've mentioned earlier, one of the things that cholesterol is very known about, and notoriously at that, is its association or correlation with cardiovascular diseases. So many inhibitors, many of the drugs that are administered to patients suffering from elevated levels of cholesterol are actually inhibitors of HMG-CoA reductase, right? And these are a comparison of the structures of mevalonate and 4 pharmaceutical compounds that inhibit HMG-CoA reductase. Right, so here's mevalonate, the natural substrate, and here are the inhibitors. Compactine when R1 is H, R2 is H. Simvastatin when R1 is methyl, this one, and R2 is methyl. Prevastatin, levastatin. I'm sure that the drugs administered to individuals suffering from such elevated levels of cholesterol are in one way or another these drugs, okay? And they target the committed step or the rate determining step of cholesterol biosynthesis, that which is catalyzed by HMG-CoA reductase, which of course makes sense. Okay. Now, atherosclerosis refers to the buildup of fats, cholesterol, and other substances in and on the artery walls. Okay. For example, here. Okay. The buildup is called a plaque, and the plaque can cause arteries to narrow, blocking blood flow. The plaque also can burst, leading to a clot. Right? This is a normal artery, and this shows an artery with a buildup of cholesterol plaque, which with resulting arterial narrowing. Okay, artery showing plaque rupture with superimposed blood clot or thrombus. Remember, I mentioned earlier that can liken the VLDL, LDL to trucks that deliver triacylglycerols and cholesterol esters to peripheral tissues, extrahepatic tissues, while the good cholesterol, HDL, as street sweepers that collect cholesterol esters, and the cholesterol esters and the cholesterol that by accident fall by the wayside. I mean, that are what do you call that? That are unloaded by accident along the blood vessels. Okay, and so they're good cholesterol because they prevent the formation of such plaques. Okay, all right. So I hope that you have learned a lot from this discussion, and that you will be able to appreciate the remarkable pathways that lead to the synthesis of cholesterol and the regulation of the synthesis of such an important but notorious biological molecule. All right? Okay. Okay.